Okay, thank you very much, and thanks for the invitation. So I'm going to talk a little bit about the work on gene therapy in the context of grafts, and it's quite topical at the moment, of course. So there may be one or two questions around clinical need. So just very brief background. We've been working on this for 20 years or so, as you'll see by some of the dates on some of the slides, but we've been persistent. Gene therapy is really an exercise in trying to deliver something useful genetically that's going to have some benefit. Usually this is in genetic disease, but of course can be in the context of cardiovascular disease, where you're just trying to overexpress or manipulate something to a useful endpoint, and that would be, for us, blocking neointimate formation in a bypass graft. Over the years, there's been a lot of hype and hope in gene therapy, and there were some problems on clinical trials. There were some very poor clinical trials that were badly designed that really impacted the field. There's been 2 deaths due to viruses on clinical trials. So the field really suffered from this. But over the last 10 years or so, and many of you probably won't be aware, there's been some really fantastic clinical data. And there are patients now alive who have genetic disease 15 years after gene therapy. So there is long-term success in certain areas, but it's not a cure-all approach that people believed it would be. And the most advanced probably at the moment, there's a phase 3 angiogenic trial going on, but there's also this CUPID-2 trial, phase 2b, which we'll report very soon, which is using SERCA2a gene transfer and an adeno-associated virus for heart failure. So this improves calcium handling in the heart. So that's going to be really pivotal for the field in general. So these are some of the examples of the very early hope. Just to put into context, this is peripheral ischemia that was magically cured by plasmids overexpressing angiogenic growth factors. But when these things, and this is the tumor that's been hit with an oncolytic virus that magically disappeared, but when these were taken through to phase 2 trials, any efficacy was gone. In terms of cardiovascular, of course, there's multiple ways of delivering. So the CUPID-2 trial, which is an AAV vector, is injected very simply into the coronary vasculature. It's a very tropic virus for the myocardium. It gets in there, starts to manipulate SERCA2a and improve calcium handling. We're interested in modifying grafts, of course, and this could either be in peripheral or the cardiac context. So Andrew's alluded to many of the mechanisms that we may target in this context, and I'm not going to talk about many of those, just to say that one of the key things that might be attractive here is the access to the tissue at the time of surgery and a clinical window where we can manipulate this with a small molecule or a virus or whatever for therapeutic gain. So this is really around about 30 minutes in which we'd have to genetically modify things, which means there's an opportunity, but also you have to have very efficient systems in which to deliver this, particularly if you need to keep the therapy localized if there were off-target effects, which is obviously very important. We've really focused most of our work on extracellular matrix remodeling and migration and proliferation of smooth muscle cells, which I'll talk about. In the context of delivery, just to very general overview, you can use non-viral vectors and viral vectors. If you have this clinical window for 30 minutes, you're not going to get anywhere with any of these DNA vectors or DNA and lipid vectors. They're just really not efficient enough. You'll maybe get about 1% gene transfer into the saphenous vein. Some other people suggest it's more than that, but I've not seen anything that's convinced me. Viruses, of course, have evolved over millions and millions of years to do a much better job. They don't come without their problems, though, because mainly they're immunogenic. This is HIV lentivirus. It's actually not particularly immunogenic. Sounds quite scary, but it actually could be a very good virus for manipulation of vein grafts. We'd like to do it clinically with this, but actually there are no companies in the world that will make enough virus that we could actually do anything useful with in pigs or humans at the moment. This is Adenovirus, which is very efficient, very easy to make, and has a high efficiency gene transfer. This is not to scale, but this is an AAV vector, which is particularly good for muscle in terms of cardiac or skeletal. So many years ago with Sarah George, when we were in Bristol, we used an adenovirus. This is adenovirus 5. Really to manipulate the vein graft. It's just very simple. Cannulation and a luminal dwell. You can do anything you like with this. You can paint it on the outside. You can bathe it. We decided to do the luminal dwell really to try and keep the gene transfer localized to the sort of subendothelial smooth muscle cell layer, which is where active proliferation and migration of smooth muscle cells was occurring. So we use a serotype 5 adenovirus vector, which is the one that's used by pretty much everybody else. It's very well characterized, but it does have one or two problems associated with it, which I'll come back to later. But essentially, if you do this 30-minute gene transfer using a high concentration of this virus, and you look a few days later, after the 30 minutes, you can just wash this free of any excess virus. But what you do get is very high levels of gene transfer. So this is the vein just cut open and pinned luminal surface upwards. Wherever you see blue, you see cells that are expressing a LAC-Z, which is a reporter gene from the virus. So this is very efficient. And if you culture that in the old human organ culture system for 14 days, so this was the original lumen of the vessel, this is the neointima that's formed, you can see that the cells that were actually infected at time 0 were at the luminal surface, but they've migrated forming this neointima. So that suggests that the virus is actually hitting the right cell population, and the virus itself doesn't have any really bad effect on vein graft kinetics, if you like, although this is clearly only an ex vivo, non-inflammatory environment. We also use the pig model that Johnny's alluded to, and Andrew's alluded to, that Johnny and Andrew developed. This is with Song Wan in Hong Kong, where essentially we can just modify the vein graft with gene transfer here and then just graft in. This is the end-to-end system that Andrew already talked about. And essentially, again, it's a very efficient system. So if you look 7 days later, then the graft itself has very high levels of this blue reporter gene. So we can actually deliver things very usefully in this system. Just one point of context, if we looked at 28 days, then this would be completely clear. The virus is very immunogenic. You get some inflammatory infiltration, but it's removed from those cells that are expressing from the virus are actually removed by the immune system within 28 days. So that puts into the context in terms of the kinetics of neointima formation across different species that might be relevant. Many people have used this system, so this is Keith Channer's work a long time ago now, using nNOS, so nNOS overexpressing using a virus in a rabbit model, essentially show that you can reduce inflammation, you can block smooth muscle cell proliferation, improve endothelial function. All those things lead to this reduced neointima formation. And Paul, who's following me, is going to talk a lot more about inflammatory aspects of vein grafting. We focused on this mechanism many years ago. So in the vessel wall, normally smooth muscle cells are maintained in this very quiescent state. After vascular injury and grafting, they're activated. You have matrix degradation, and these metalloproteinase enzymes seem to be activated. They lead to the ability of these smooth muscle cells to start moving, start proliferating. So the very simple strategy was to overexpress inhibitors of these proteins, and these are tissue inhibitor metalloproteinases, really to shift the balance back to an anti-migratory, anti-proliferative phenotype. So we put the virus together with one of these TIMPs to see what would happen. And to cut a long story short, these are very old papers now, but we worked out exactly what was going on in cell culture, but also in vivo. So we genetically engineered a virus to overexpress TIMP3. This gets into smooth muscle cells relatively easily. The receptor actually for the virus isn't really expressed on smooth muscle cells at all, so you have to force it in using high concentrations. And then from the nucleus, it uses all the DNA machinery to express whatever you put in. TIMP3 then has a signal peptide on it. It's secreted. It binds to the matrix around these smooth muscle cells. And then through its ability to block metalloproteinases and also metalloproteinases like family members, it reduces smooth muscle cell movement and invasion. And at high concentrations, it can actually kill those cells. It sounds like a really bad thing to do, but this only really happens at very high concentrations, which is at the subendothelial layer. So this actually combined approach might be quite important. And this is just some of the early data in the human organ culture model. This is the control. After 14 days, you have this neointima formation. Of course, this is an absence of inflammation, blood flow, or anything like that. It's a very simplified system. But if you overexpress these inhibitors, so this is TIMP1, TIMP2, or TIMP3, you can actually block this neointima formation, suggesting that if you do block movement and proliferation of smooth muscle cells, you can do something quite useful. And in the pig model, this also worked. So we published a 1-month study in 2000, and then we did a 3-month study, which somehow took us 11 years to publish. But essentially, the regulators really wanted us to do this 3-month study to see if we got a sustained effect. There's a lot of evidence in animal models, actually, that if you block early migration and proliferation, you have a catch-up effect. So they wanted to see whether this was an issue, but it's not, because at 3 months, you still get inhibition of neointimer formation. As I mentioned, the important things here are that the virus has actually disappeared from the graft at 3 months. It's gone at 1 month. So blocking early remodeling seems to be quite important in this model. Human relevance is to be assigned, but in this pig model, if you do this thing early and block at the time, short times after grafting, you get this long-term effect. The other important thing to note is if you're using a control virus, which is this RAD60 virus, that doesn't express the therapeutic gene, you have no detrimental effect. So putting a complicated virus into saphenous vein context has no detrimental effect. So that's an important safety feature of this virus. So based on that, we are funded to do a clinical trial by the MRC with co-funding from the British Heart Foundation, which is originally 2011 to 2016, but I think it'll be 2011 to 2018 at least, where we need to move this virus into GMP, produce a clinical grade virus, get the regulatory side sorted. That should be relatively easy. There's nothing complicated about this virus. It's the first in man, but only for the therapeutic gene. Phase 1, dose escalation, make sure that at the time of surgery, the slight modification of doing the gene transfer isn't detrimental. And then phase 2 design, should we ever get there, would be patients who are having 3 grafts, a main arterial graft, and then the other 2 would be randomized either to placebo or gene therapy. So it's within placebo-controlled trial. The power calculation at the time was about 120 patients looking at OCT imaging for neointimer formation at a year. So we are in the process of going through this. We've had an enormous problem that's taken us 2 years to solve in making the clinical grade virus, and I'm not going to bore you with it. But essentially, when you take things from the lab and put it into the clinical grade, you're taking a very small tube of virus and you're making a bioreactor full of this. And I told you that TIMP3 is quite a gene that can affect proliferation, it can affect lots of different things. And the virus is so clever that during this production, when you amplify the virus, it seems to mutate it, mutate TIMP3 so it doesn't express, and that allows the virus to replicate much more. So we can't use this system at all. So we spent a year working out how we can do it, and we can use a TET repression system in GMP compliance to do this. And there's one source of that cell line in the world that will allow us to do that. So we'll put that into place. It's going to cost about 1.5 million to make the virus. So this actually, at the moment, the trial is on hold by the MRC until we get approval for the extra finances to make the virus. And also the MRC are, this may be important for all of you, are reassessing whether there's a clinical need for new therapies in vein graft bypass surgery using saphenous vein. So we shall find out what the MRC think next week. Just to go back to the virus for a minute, there are a couple of important features that I think are relevant. Everybody uses this virus. We know an awful lot about it. And in terms of using it as a bypass graft therapy, its tropism, its infectivity for the graft tissue is actually pretty poor. You have to force it in. 80% of people in this room would have antibodies against the virus. So that may not be important when you're doing ex vivo gene transfer. But when you graft in an Ad5 transduced vessel, you may have some complications. And the fact is the virus only hangs around for a short time, which is okay in a pig, but who knows about that in a human. So one thing that we do is work with different types of viruses. So this is just a phylogenetic analysis of adenoviruses, human adenoviruses, and everybody works with this one pretty much. But there are actually about another 60 of them that are out there in nature. And nobody really knows much about any of them, apart from people who work in the vaccine world. So people who are working on Ebola vaccines, HIV vaccines, are using many of these human and non-human viruses because they're incredibly useful for vaccine due to their properties. But we can also use these beneficially for gene therapy, and I think in the future we may want to think about how we use some of these effectively in this context. I'm just going to show you one that we're particularly interested in. So this is adenovirus 49, which sits over here somewhere. It's a subgroup D virus. There are 2 papers ever published on it. There's nobody with any, nobody's ever seen this virus, and nobody has any neutralizing antibodies against it. So we'd wipe out any of the immune problems. So this just shows you in Glasgow, if you look in Glasgow cardiovascular clinics and take serum, about 30% of people have neutralizing antibodies against the virus. In the general population in Africa, America, places like that, it's high, it's about 80%. But AD49, there's actually no pre-existing immunity against AD49, so that's a pretty good property of this virus. And actually, when you combine that with its infectivity properties for primary smooth muscle cells, which are our target, it's incredibly good. So you just do this very simple experiment. You expose the virus or the control AD5, which we all use, smooth muscle cells, different doses. These short clinical time periods, so exposing cells just for 10 minutes, 20 minutes, 30 minutes, or an hour, you get very high levels of gene transfer. So we can boost the infectivity into the bypass graft as well as eliminate the immune consequence at the same time. So we think this may be quite a useful virus in the future. And if you use a GFP version of this virus, this is just looking at bypass graft tissue, you can see you have very nice transduction. This is actually endothelial on this particular one, but you have very high levels of gene transfer at pretty low concentrations of this virus. And just a final thing that may be useful in the future, clinically maybe a bit more challenging, but David Dechek, who works in Seattle, has recently published that one of the things, if you transduce, this is rabbit jugular vein grafts, if you transduce with AD5 and leave it in culture, this level of infectivity stays, of course, because there's no immune response against the virus. But if you graft it in and just look 3 days later, you do lose a lot of the gene expression very early on. So that's probably not going to be particularly useful in the clinical setting, although, of course, it works for us in the pig. But his concept is to do 2 things. One is to manipulate the genome of the virus. And you take the genome of the virus out, you lose some of the immunogenic properties of the virus. Quite difficult to make, but these are so-called helper-dependent viruses. And the other thing he has done is go back in and not transduce the graft at the time of surgery. He's done it 28 days later. So he's gone back in vivo with a catheter approach to manipulate the graft. And so when he does that, he gets low level but very long-term expression from the virus. So that may be useful. The caveat to that clinically, of course, is you have to go back into the patient 28 days later, and you might have missed something that's really important. But who knows? It's potentially an opportunity, but I can imagine clinically quite challenging. So just to summarize, I think gene therapy with TIMP3, we may get there, depending on what the MRC say, and our ability to make this virus ever at clinical grade. Adenoviruses really are very effective, so they're a very useful tool to dissect mechanisms as well as potentially clinically useful. Will they last long enough? We don't know until we actually do a trial. There are many strategies to improve the properties of viruses to make them optimal for cardiovascular gene transfer, particularly in the context of bypass grafts. And any of these modifications that I've talked about might further improve that. But I think the first thing to do is actually do the trial with the traditional AD5 vector and see what we get. And just to thank people who've been involved long term with this work, Sarah George, Song Wan, Andrew Newby, and everybody in Bristol. And these are some of the people I work with in the European Commission funded projects who are identifying all of these strange other human and non-human adenoviruses. And thank you for your attention.